Okay, good afternoon, everyone, and welcome to the front row. My name is Jamie Williamson, and I'm going to be your host today when we're going to hear Ali Torkamani talk about coronary artery disease. Before we have Ali come up, I want to say a few things and give a sort of a general introduction. We're kind of in this area where we're moving away from a generalized approach to medicine and moving more into the era of individualized medicine. And Scripps is a great center of innovation in this regard. So in the past, basically physicians and scientists, just focused on the average behavior of a very diverse population of individuals. And in fact, clinical trials were designed to make sure that nobody had an adverse reaction. And whether the drugs were appropriate for a diverse population or not was not part of the design. But advances in genomics have made it possible to characterize people and drugs and their reactions to drugs using all of these genomic techniques. We can look at the DNA, we can look at the proteins, we can look at the RNA, we can look at the immunological response, and now we can look at the microbiome, and that affects everything. So the center here that's really innovating is the Scripps Translational Institute. And that's the site, this is Eric Topol's institute, that's the site that has the All of Us grant. And their goal is to enroll 1 million patients and keep track of them on an ongoing basis with all of their health records, their genetics, and then try to use this information to better tailor individualized medicine. There's also a very diverse set of investigators developing what we call digital clinical trials. And so these are not just located at a hospital, but are diverse and usually involve some app-based interaction between people that enroll in the programs and the institute to try to share information between people facing common problems and also provide them best advice on how to manage their issues. And there's a variety of things, and this is a great, you can look at their website that you can enroll in these things, but there's nutrition, there's infectious disease, maternal health, so power mom, sleeping, and of course, heart health, which is the role of today's talk. Now, so we're talking about precision medicine, and I think everybody had some feeling, even some of us who are older, about risk stratification. At a certain time, everybody knows somebody that said something like this: well, my grandfather died of a heart attack, my father died of a heart attack, I'm probably going to die of a heart attack. And that's not a very good risk stratification, because it doesn't take into account genetics, and it doesn't take into account your environment, your behavior, exercise, and diet. So the thing that's really changed is that now you can sequence your own genome. And then the problem advances to a different point. Well, now you have the gene for X. And one of the questions that Ali is trying to help people address is, well, what do you do with that information? And so that's called risk stratification. So you need to know, are you at risk? And then what should you do with that piece of information? And so he's going to be telling you about app-based delivery of genetic risk scores. And this is really the intersection between genomics and medicine, machine learning, and there's a bit of psychology involved in making this work. So one of the major tools is artificial intelligence or machine learning. And I think most of us hear about the great concern about machine learning. And this is largely centered around these large language models and the potential for abuses and disinformation that's harmful to society. But at the heart, machine learning is a very powerful tool that can take in a lot of information and recognize patterns that humans can't see. And then we can use that power of that learning to make predictions. And that requires a lot of data. So you need millions of patient records in order to train one of these AI models to make some prediction. And so that's what Ali is going to be talking about. So I think the goal of this is to try to provide you a tool that better informs what you can do with the information that you have about your present health state and your genetics to make informed medical decisions. And we'll probably talk at the end about how you engage your physician in those discussions. So I'm going to let Ali introduce himself and tell you a little bit more about where he came from and how he came to be at Scripps. He'll give his presentation, and then I will come up back to the stage. We'll sit over here, and we'll have a little discussion. So we have many people that have logged in virtually, and you can type at any time a question that you might have into either the chat or the Q&A. And then for those, we have a nice group of about 100 people here in person, and there will be microphone runners, so put your hand up, and I'll try to get as many questions as I can, and we'll have a really good discussion, because I know this is a topic that's generated, it's very personal and a lot of great interest. So without further ado, please welcome Ali.
All right, thank you, Jamie. So as Jamie mentioned, I'm Ali Torkamani. I'm going to be talking about preventing heart disease with machine learning and smartphone technology. But before we get into that, say a little bit about myself and how I got here. My interest in genetics and machine learning really derives from a lifelong interest in genetics, I would say. I've always been interested in genetics as far as I can recall. And that, in turn, is derived from an interest in questions about freedom of will, actually. So I've always been interested in this concept of if we could measure everything about a person, everything about their current state, their past, what they've done, what their current status is, and perhaps we could make a prediction about what the next thing is that they are going to do. And if that is the case, then do we really have free will, and what are the ramifications of that in terms of, our purpose in life and that sort of thing. So I bet you guys were not expecting to get a dose of existential dread in a genetics and machine learning talk, but that is really where it's derived from. But then, from a more practical perspective, I thought, well, genetics underlies a lot of, or at least a big portion of the information that you might use to make such predictions. So maybe we can do something more practical with that. So I got my bachelor's degree at Stanford in chemistry, then came to UCSD for my PhD, where I got more into the computational biology and genetics, and then made the move to Scripps right at the time that Eric Topol was starting the Translational Institute, and sort of worked my way up from there. So I spent essentially all of my career developing computational tools for genome interpretation broadly, meaning if you have someone's genetic data, what can you tell them about that? And genetic data is very large. You need computational tools essentially to process it and understand it. And so in the early part of my career, I developed tools for interpreting genomes from a rare disease perspective, and discovered a couple of rare diseases, actually characterized a couple of rare genetic diseases. And as time went on and data sets got bigger, then we're able to actually look at very large populations and combine genetic data with other types of data, biomarker data, EHR data, behavioral data, et cetera, and then use that to make future predictions about a person's disease risk and what you can do to intervene on that risk. So that'll be the focus of my talk now here. All right, so the talk outline. First, I'm just going to introduce you to heart disease or coronary artery disease. What is it? How can we prevent it? And why are we failing? Then I will describe genetic risk at a basic level. What is it, and how can we use it to improve prevention? Then get into the delivery of that genetic risk information to get people to improve sort of their approach to prevention? And then finally, how can we amplify the benefits of genetic risk using machine learning? All right, so what is heart disease or coronary artery disease, and how can we prevent it, and why are we failing? Heart disease is the leading cause of death in men and women in the developed world. You see here, in terms of what kills us, heart disease and cancer are essentially neck and neck in terms of causes of death, but heart disease is largely ignored by most people. So this is what the media covers, proportion of sort of mentions of different causes of death in the media. You see heart disease barely registers up here. It's mostly about more dramatic sort of events. And then that trickles down to what we end up Googling, what we're interested in learning about. Again, heart disease barely registers at the top of the plot here. We're spending most of our time thinking about cancer, which I get it. It's scary, but you're just as likely to die from heart disease as you are from cancer. All right, so what is heart disease or coronary artery disease? It's essentially the buildup of plaque or lipids in the coronary arteries of the heart. That leads to a lack of blood flow. It can eventually lead to a complete closure of the coronary arteries or blockage with a clot, and that ultimately leads to a heart attack or stroke and potentially death. The risk factors for heart disease are shown here on the right side of the slide. And those risk factors are lipids that circulate in your blood, these cholesterols here, lifestyle, sort of a lack of an active lifestyle, unhealthy diet, smoking, being a man is associated with risk for heart disease, high blood pressure, diabetes, obesity, and of course, genetic factors. How can we prevent coronary artery disease? It's pretty simple. You control the risk factors. So you control your blood pressure, don't smoke, sleep properly, eat well, be active, control your blood sugar, manage your weight, and control cholesterol. Pretty simple, I would say, in terms of words, but maybe harder to put into practice. All of these things are, many of these things are under our control, with the potential exception of cholesterol. Actually, most people think that you can reduce your cholesterol levels through a healthy diet or activity, and that's true to some extent, but cholesterol is under genetic control to a large degree, and in some individuals, it needs to be lowered with medication. But we're doing a pretty poor job at that. If we just look at control of cholesterol on its own, for people that are indicated to take a statin based on current clinical guidelines, only about 30% of those people actually adhere to taking a statin to reduce their cholesterol levels. And these are the reasons why. The primary reason is worry about side effects, and then after that is, you want to try diet and exercise first, don't think you need one, don't like medication, prefer natural remedies, etc. So all this really boils down to, I think, is a kind of a mental cost-benefit analysis. We're already sort of ignoring heart disease in the first place, so the risks of heart disease seem pretty low. So in that light, maybe it doesn't make sense to take a medication when you do that sort of risk-benefit analysis on your own. If we're not very aware of the risks of heart disease, then we're unlikely to intervene on that risk. If you were to follow guidelines, the benefit from reducing cholesterol sort of breaks down as shown on this slide. So on this side of the slide is If we ignore genetics and sort of follow guidelines as they are right now, when you go to your physician, they essentially calculate a 10-year risk of you having a heart attack using a standard clinical calculator. And if your risk is above a certain level, these two levels highlighted here, then you're clinically indicated to initiate lipid-lowering therapy or reduce your cholesterol levels. And again, only 30% of people that actually meet these thresholds are actually reducing their cholesterol levels. In that scenario, you have to treat about 50 people with lipid lowering to prevent a single heart attack. And you don't know, among these 50 people, we don't know exactly who's the one that's going to have a heart attack, so you need to treat all 50 people, one of which will have a heart attack that would be prevented by the treatment. So perhaps another reason why people are not so enthusiastic about taking lipid-lowering medication. But you can change that with genetic risk. So you can stratify people into high and low genetic risk, and I'll get into in a bit how you actually do that. But if you were to stratify the population into high and low genetic risk individuals, then you see that among these groups, there's a big difference in what's called the number needed to treat, or the number of people that need to take a lipid-lowering therapy to prevent a heart attack. So among the high genetic risk individuals, you need to treat about 20 individuals to prevent a single heart attack. For average genetic risk, it's about the average of the population. And then in low genetic risk individuals, that number goes up to 60 to 1. And actually, under some sort of estimations, this group may not actually get much benefit from lipid-lowering therapy overall. And later on, I'll get into why that might be the case. But anyway, we can use genetic risk to stratify the population and perhaps identify those people that are both at the highest risk in terms of their genetics and those that would get the greatest benefit from actually intervening on their risk. All right, so how do we actually identify these high genetic risk individuals? How do we stratify people by their genetic risk? So first, I'll describe the basic sources of genetic risk. Jamie in his introduction mentioned you have the gene for X. That is a way of describing monogenic risk. Monogenic risk means mono being one, means that this is risk driven by a single genetic variant. You have a single genetic variant. It drives very high risk for disease and commonly referred to as the gene for X. So BRCA is an example of that. I think probably a lot of you are familiar with Angelina Jolie having a double mastectomy due to having the gene for breast cancer. She had a BRCA mutation that she intervened on with a double mastectomy. This sort of genetic risk is very rare. Very small proportion of the population has monogenic risk for a common disease. In the coronary artery disease space, it's familial hypercholesterolemia would be this monogenic risk, or mutation in a handful of genes that leads to extremely high cholesterol levels that increase your risk for heart attack. And even among these individuals, I've spoke to some individuals that have familial hypercholesterolemia, and even among these individuals, you'll hear people that say, well, I want to try to reduce my cholesterol levels with diet and exercise, and it's basically impossible. You cannot overcome that level of genetic risk with diet and exercise. On the other side, there's polygenic risk. Polygenic risk actually underlies the majority of genetic risk for most common diseases in the population. It's a lot more complex than monogenic risk. It's the accumulation of many genetic variants that are common in the population, and each of which contributes a small amount of risk to you, and it's the accumulation of those genetic variants that lead to high versus low risk individuals. Here's how we generate those polygenic risk scores or genetic risk. In short, it's derived from data from essentially millions of individuals profiled genetically. So you start with a study where you're genomically characterizing the genomes of a million plus individuals these days, and looking across all their genetic variants. So this is an image of chromosomes splayed out on the x-axis. Each dot here is a genetic variant. The y-axis is the degree to which it's associated with the trait of interest, let's say coronary artery disease. So the higher these peaks, the more associated that single variant is with risk. You identify those that exceed a threshold of risk. So you have this handful of genetic variants that are associated with risk for coronary artery disease. And then when you look at a new individual, you can sequence their genome, look at these sites where there is a known risk, and essentially add up their risk across those sites. So each one of these sites from this study is going to be associated with a weight, how much it contributes to your risk. Then you look in a single individual and essentially add up those risks, and you get a polygenic risk score out of it. Those people whose genomes are enriched with genotypes that put them at risk would be at the right end of the distribution. So they have a high score, which puts them at high risk, versus the individuals at the lower end. Their genomes would be sort of depleted relative to average with these risk variants. So they're at low genetic risk. And this is how you can sort of stratify people into high and low genetic risk in a more complex manner. And I think it's probably obvious from what I'm sort of led with, the genetics is often not deterministic. So a lot of people think of genetics as fate. If you're high genetic risk, that's it. There's nothing you can do about it. No, that's not the case. You can intervene on your genetic risk in a number of different ways. I've reviewed it in detail in this publication here. But you can inform therapeutic interventions in people at high risk, like the lipid-lowering therapy that I'm going to discuss in a bit. But there's other uses of this information as well. You can inform disease screening. So in cases like, say, breast cancer, prostate cancer, that sort of thing, you can, say, accelerate screening in individuals of high risk, or maybe decelerate screening in individuals of low risk. And even when there's nothing you can do from a screening or prevention perspective, you can even do sort of life planning. So Alzheimer's disease might be an example of that, where there's not much you can do about it, although now there are actually therapies approved for Alzheimer's disease. But you can think about life planning for the future if you're at risk for some of these other conditions. And we know that polygenic risk is important for sort of healthy living or having a healthy lifespan. We've shown this in another study that I'll briefly mention, called the Welderly study, where we profiled the genomes of about 1,000 individuals over the age of 80 with no common chronic diseases. So they reached the age of 80 and they didn't have a heart attack or cancer or any of the common conditions that would put people down. And when you characterize their genomes and calculate these polygenic risk scores, you observe that they have lower polygenic risk on average for coronary artery disease and Alzheimer's disease. So their genetic makeup is at least part of what makes them a Welderly individual. So it has real-world impacts on health and longevity. All right, so with that, I'm going to get into the delivery of genetic risk information. So can we use this genetic risk information to actually drive the initiation or adherence to preventative therapies? And we've pursued this question with two different studies. I'm going to be focusing mostly on this MyGeneRank study. So we've developed 2 smartphone-based approaches to deliver genetic risk information to people and see essentially what they do with that information. So the first is the MyGeneRank study. It's you can access the app here at mygenerank.scripps.edu. It's open to anyone with basic inclusion criteria. Basically, you have to have a phone, you have to be an adult, and you have to have a 23andMe profile. So you need to bring your own genetic data for this study. And if you sign up, I'll go through what you need to do, but eventually you'll get a genetic risk score that you can act on as you please. And then we have a second follow-up study that we've been working hard to launch. It's a closed study, so it's all by invitation only, and it is essentially a randomized trial to prove some of the observations that we've made in the MyGeneRank study. So this is what the PEPPERS app, or Prospective Electronic Polygenic Risk Study, this is what the app looks like. The welcome screen, there's sort of a list of tasks that you need to complete for the study, then 2 views of genetic risk, one being just genetic risk on its own, ranked on a 0 to 100 scale, and then a second view that integrates genetic risk with clinical risk, cholesterol levels, age, gender, whether or not you're diabetic, et cetera, to give you an overall risk. And then you have the ability to change those risk factors, like, for example, lower your cholesterol level in silico in the app and see how that changes your risk overall. So we'll calculate a new risk for you based upon any factors that you want to change within the app. That'll show you what your new risk will be, and the idea is essentially for individuals to use that information to make decisions about how they might want to reduce their risk. So we never tell anyone what to do in either one of these studies. We just present their risk and provide them with tools to see how they can modify their risk, and it's up to the individuals to determine what they want to do with that information. Other aspects of this app, it's completely electronic, so you don't have to see anyone to participate in the study. You can go through the inclusion criteria and informed consent fully within the app itself. We have some privacy-preserving technology and some other technology so we can collect EHR data, mobile health data, send people to get testing, and then these interactive results, and we deliver surveys essentially to individuals to get their response to that genetic risk information. So we've delivered that information to thousands of people with MyGeneRank, and then measured what they've done with that information. So I'll get into that now, the MyGeneRank study and the results of real-world delivery of genetic risk information. The design of MyGeneRank, as I mentioned, is meant to be sort of a self-efficacious design. People sign up for the app on their own. They provide their own genetic data. They link up their EHR data on their own. It's straightforward. It's essentially logging into your patient portal. We prompt you to do that, and that provides us access to your EHR data with your permission. And then they can play with the app, educate themselves on their own. And it's also up to the individuals as to whether or not they want to interact with their physicians or share this information with their physician. When we launched this study, the app was not as nice as the prior app that I showed you. So this is actually what it looked like at launch, and what the results that I'll show you are based upon. A relatively messy view. Cleaned it up a little bit later on. It's available in Spanish as well. There's virtual genetic counseling integrated as well. So there's a sort of online genetic counseling service that you can access via the app. And if you were to download it now, it'll look more something like this. So the results will be based on this view, but the current view is more like the apps shown on the right, screenshots shown on the right. We've gone through a large number of experiments to essentially improve the understandability of the information. So this view is meant to improve how people understand the information that's being delivered to them. Okay, so this is how the study actually works. People download the app, they join the study, provide their informed consent, link their genetic data, and then they do a baseline health survey, a health survey that essentially asks them about their health status and whether or not they're taking any medications. A genetic risk score is calculated, it's returned to them within the app, and then immediately after they get their result, they'll get a psychosocial survey. So this survey is more about your feelings and kind of emotional response and understanding to the genetic information that was just delivered to you. And then at any time 6 months after the delivery of that information or later, they're provided another follow-up health survey that's very similar to the baseline health survey, essentially repeats those health questions, and we use that to determine how people have changed their behavior before and after receiving their genetic risk information. We also did an email-based follow-up as well. And cutting to the chase, here's what we found. So if you deliver genetic risk to individuals and then determine who decided to start taking lipid-lowering therapy, we found that among high genetic risk individuals, they were more than 2 times as likely to initiate lipid-lowering therapy than the low genetic risk individuals. And not only that, they're accelerated the initiation of lipid-lowering therapy by 10 years. So if you look at the average age of the high genetic risk individuals who initiated lipid-lowering therapy in response to this information, they're about 55 years old on average, whereas if you look at the low genetic risk individuals that initiated therapy, they're about 65 years old on average. So you get increased initiation and earlier initiation of lipid-lowering therapy in people who are told that they are at high genetic risk, which is exactly what you would want to see for prevention of heart disease in these individuals. And then what about the psychosocial response? To gauge this, what we did was deliver some statements, some of which are up here, to these individuals within the survey, and they're asked to agree or disagree with these statements on a 1 to 5 scale, 1 being strongly disagree, 5 being strongly agree. So you see here, my genetics make it more likely that I will develop coronary artery disease. People with high genetic risk more or less strongly agree with this statement. People with low genetic risk disagree with this statement. So you have a nice relationship with their agreement respect to their genetic risk, essentially showing that people understand their results. People that are high genetic risk agree with this statement. People with low genetic risk disagree with this statement. One thing that is often stated to detract from the use of genetic risk information is that it makes people worry for no reason. That you're giving information to people that's just going to make them anxious, and there's nothing they can do about it. I mean, that's wrong anyway. There is something you can do about it, but we also see that there's no undue anxiety as well. There's a slight association with genetic risk, but overall these people are around a 3 or 2, meaning neutral or disagree. So overall, they are neutral or disagree with the statement, I worry a lot about developing coronary artery disease. So no sign of undue anxiety. And then in terms of self-efficacy, I am able to reduce my risk for developing coronary artery disease. Everyone uniformly agrees that they're able to reduce their risk for coronary artery disease. So they feel empowered, they're not worried about it, and they understand their genetic risk. We're also ignoring useful genetic risk information in this study. So we're really only talking about high genetic risk individuals, but what about the low genetic risk individuals? Is there something different that they could do? And I think that is potentially the case. So we saw this before. all these individuals are clinically at high risk for coronary artery disease, but only these people are at high genetic risk. These people are at low genetic risk. So it sort of begs the question, why are they at risk overall? It might be due to lifestyle rather than genetics. Your risk overall is essentially a sum of your genetics and your environment. If your genetics is not contributing to your risk, then it is likely your environment. So perhaps for these people, a lifestyle intervention is more important versus in these individuals, a therapeutic intervention is important. These people need to offset genetic risk, which in many cases cannot be done through lifestyle changes. These individuals don't have genetic risk to offset. They have lifestyle risk that they need to adjust. All right. And then moving on to the PEPPER study, what do we need to do to prove this out and really drive broad adoption? The PEPPER study is designed to answer these key questions here. Does the genetic risk information change physician decision making? So we understand it from a patient or individual side, but we haven't measured it on the physician side yet. Do the changes in preventative behaviors that we observe, are they persistent? Do the people continue to adhere to medication, or does that drop off over time? We need to answer that question. And ultimately, are health outcomes improved as a result? So does the uptake of lipid-lowering therapy ultimately result in a reduction in heart attacks in the long term? And so to do that, we've designed the PEPPER study. It is a 2-phase study, 1,000-person study that is very similar to the MyGeneRank study, but now with objective outcomes, EHR-based outcomes, so actually diagnoses and medication prescriptions that we measure from the EHR rather than from survey, and with longer-term follow-up, so we can look at adherence and persistence of the use of those medications. And then there will be a second phase, a 10,000-person randomized study where some people will get coronary artery disease risk, some people will get risk for glaucoma as control with longer-term follow-up, so we can actually observe whether or not this information reduces heart attacks. This study is generously supported by the Volo Foundation. There are other opportunities for expanding these studies as well. So I'll very briefly talk about breast cancer. In breast cancer, I mentioned the BRCA genes. These people have about 50% chance of developing breast cancer versus the normal population. In a woman's lifetime, you have about a 12% chance of developing breast cancer. And under these sort of conditions, women are told to start screening at the age of 50 for breast cancer. But it turns out that if you use genetic risk information combined with non-genetic risk information to predict risk for individuals, and then use that information to stratify when people should get screening or mammography screening, you can decelerate or accelerate screening in a pretty large proportion of people. So it turns out that if you follow guidelines and start screening at 50 years, in 10 years, 40% of women will be called back for additional imaging due to a false positive result, and 5% of women will get a false positive biopsy of their breast. If you use genetic information and non-genetic information, 16% of women at the age of 40 would have the risk of an average 50-year-old woman. So those individuals could accelerate their screening starting at 40 years old rather than the recommended 50. And 32% of women at 50 years old have the average risk of a 40-year-old woman. So they could wait 10 more years to initiate screening since they are at a risk that is equivalent to that of a younger woman. So this is another opportunity to take screening and stratify it by risk rather than just telling everyone to start at the same age. And actually, if you are already signed up for MyGeneRank, you will very soon be able to get your breast and prostate and other polygenic risk scores as well. So we're just about to roll this out. You'll get if you're already signed up, you'll get an email essentially asking you to opt in to receiving these other genetic risk scores. So go beyond coronary artery disease. All right, so with that, I'm going to spend the rest of the time sort of briefly talking about how we can amplify this further. A polygenic risk score is a pretty simple formulation of genetic risk. We can do a lot better with machine learning, and I'll show you what we're doing there. I want to mention that a lot of this work is the work of Sean and Hossein in my lab right here in the row over here. What we've done is develop a genetically informed approach to meta-prediction. What does that mean exactly? We're using 2 levels of prediction, essentially, to make predictions about a person's genetic risk. As I mentioned previously, the risk factors for coronary artery disease include type 2 diabetes, smoking, sex, etc., behaviors. So to power this meta-prediction approach, we make predictions about not only development of coronary artery disease, but development of all those contributing risk factors as well. So for type 2 diabetes or other diagnoses that drive your risk for coronary artery disease, we predict whether or not you should have had an early onset diagnosis, a late onset diagnosis, or any onset diagnosis prior to the point that you are now. So there's some individuals that maybe their EHR record doesn't state that they are diabetic, but they perhaps are diabetic. So we make that prediction using their genetic data and any other data available to you. We also predict their modifiable factors, so cholesterol levels, blood pressure, whether or not they smoke, whether or not they drink. We predict that from their genetic data plus other data. It turns out that many of these things are somewhat predictable using genetic data. And then we also predict future diagnoses. So we predict whether or not you might be diabetic in the next 10 years, or might be diabetic in the next 20 years, or might have heart failure in the next 20 years. And we combine that all together with your actual measures of those factors, so whether or not you're diagnosed with diabetes or not, the prediction as to whether or not you should have been diagnosed with diabetes or not, your current cholesterol levels, what we predict your current cholesterol levels should be, and all that gets combined into a final meta-prediction model. This is Sean. This is Sean on a hike. All right, so how do we actually do this? It requires a lot of data. So we have the genomes and phenomes of 500,000 individuals from the UK Biobank. We stratify that into prevalent cohort, meaning individuals that already have had coronary artery disease, and a cohort of individuals that will have coronary artery disease over the next 10 years after their baseline measurements. We use this prevalent CAD cohort to make all those contributing predictions that I mentioned. You're predicting whether or not they have diabetes at baseline or not, what their cholesterol levels are, what their lifestyle is, whether or not they might have diagnoses in the future. And we take those models, make predictions within these individuals for all these factors, combine them with their actual measurement of those factors. That goes into a final 10-year model for the prediction of coronary artery disease. And then once we have that, we can take any person's data, run it through this model, get an explanation of what their risk is, and then predict whether or not they're going to have a heart attack and what interventions would be most optimal for reducing their risk. Besides the sort of machine learning and data science side of things, the work is also informed by a large number of physicians that I work with at the Translational Institute, listed up here. So this is a sort of a summary of all the different features that go into this predictive model, some of which are selected in a computational manner, some of which are selected by the input of our expert physicians here. So what ends up being the most important for predicting risk of coronary artery disease? It happens to be the models that I described for predicting past events from genetics. So this is a ranked list of the different features that were important for the prediction of coronary artery disease. The ones with this green arrow pointing to the left, those are models that predict whether or not the person should have had one of these diagnoses in the past. So it's not that they had revascularization, which is a surgical intervention for coronary artery disease. It's a prediction as to whether or not they should have had revascularization in the past. And this shows you what the predictive features are that went into this model, and they're basically all genetic risk scores plus sex and age. So we're predicting here whether or not the person should have had surgery based upon their sex, age, and a series of genetic risk scores. And that's similar for this other prediction of the past, and this is actually a prediction of a future surgical intervention. And you can see here the prediction of the future intervention contains genetic risk information, but also contains information about their current state, their waist-hip ratio, whether or not they're taking any therapies, et cetera. And then on the right side of the plot here, these are other predictions that are not directly related to coronary artery disease, but are useful for predicting coronary artery disease, like their white blood cell count, their waist-hip ratio, whether or not they'll have an aneurysm, whether or not they have or are predicted to have cardiomyopathy. So the most important features, the point is the most important features are our predictive models. The meta-prediction model really drives the power of this predictor. And this is what it looks like relative to other scores out there. So I mentioned physicians will calculate this 10-year risk of a heart attack. That's called the pooled cohort equation in the United States. In England, they use a different score called the Q-risk3 score. And these two blocks are other attempts at using genetic risk information to make this prediction, but not in a meta-prediction model. Here's our model here. This point here is the top 1% of predicted risk. You can see within these individuals, if we predict you're in the top 1% of risk, you have a 60% chance of having a heart attack. So when we look at whether or not our predictions are right, 60% of these top 1% people actually had a heart attack within the next 10 years. But if you use the pooled cohorts equation, it's about 20-something% of the top 1% have a heart attack. So, we're more than twice as predictive than the tool that is currently used by physicians, and that's true for the English score as well as these other genetic risk scores. If you look at it over time, so you can see over 10 years, what's the sort of accumulation of heart attacks? At about 6 years, the top 1%, 50% of them will have a heart attack. When you look at these other standard scores, top 1%, at 6 years, about 20% of them will have a heart attack. So we're doing a much better job identifying people that are going to have a heart attack in the future. Why are we doing better? I'm not going to describe what's going on in this plot. It's just a way of comparing our tool versus these other tools using a metric of accuracy. Ultimately, what this is showing is that our tool is better able to predict who is going to have a heart attack in individuals who are younger, who are 55 years old or younger, who are typically considered low risk, or people that have low PCE and Q-risk scores, those clinical scores. If they have a low score, they're considered low risk overall by clinicians, but we're able to pick up events in these low risk individuals. And then finally, we're going to get into, in the last bit, what can you do about that risk? Here we have plotted in the different colors, different targets for lipid reduction, different targets for reducing your diabetes risk or your glucose levels, and different targets for reducing your blood pressure levels, and that's plotted against genetic risk. The fact that these lines go from basically top left to the bottom indicates that for people at higher genetic risk, hitting these targets leads to greater benefit than it does for people at low genetic risk. So these low genetic risk individuals, they are not getting much benefit from hitting these cholesterol targets versus the people at this end of the scale, high genetic risk individuals get enhanced benefit from these various interventions, and it's related to the genetic risk of the corresponding analyte. If you have high type 2 diabetes risk, you get more benefit from intervening on that risk. If you have high blood pressure risk, then you get more benefit from intervening on that risk than the low genetic risk individuals. And finally, we can identify subgroups of individuals that have different profiles of their benefit from clinical interventions. And I should state that we didn't train the model to predict interventions here. All we're doing is changing the values of, changing the cholesterol levels, let's say, of individuals and measuring their predicted risk that comes out of the model. So the model is automatically learning that higher genetic risk individuals get more benefit from the corresponding interventions. So we clustered individuals by their risk. We identified 5 different subgroups of risk. This set of figures here relates to targeting cholesterol levels. Again, this relates to targeting your diabetes biomarker and blood pressure marker. And essentially, you can see that there's different profiles here. So the red group gets greater benefit from lipid-lowering therapy, but not much benefit from intervening on their blood pressure. These green and purple groups get more benefit from intervening on their diabetes risk than they do from their cholesterol risk. And that all goes back to the fact that they have different genetic risk profiles, and those genetic risk profiles mediate the benefit that they get from standard clinical interventions. So with that, I'll recap the talk. Coronary artery disease is deadly, and we don't care. Genetic risk can find the right people who should care. Genetic risk can convince those right people to care and do something about it, and we can do even better with machine learning. This work is the work of a large number of people within my lab, including interns, funding. I'd like to call out specifically the Volovoud Nation, the Kessler family, and the Stowers Family Foundation. And of course, it's driven by our Center for Translational Sciences grant that allows us to interact with physicians as well. Thanks for your time.
All right, thanks, Ali. Let's have a little discussion. I wanted to start out by, so to prepare for this talk, right before I came over, I sat down and asked chatbot GPT if I should take a statin. And it said that I should consult my physician.
It's the right answer.
That's right. And so I did want to make the point that neither Ali nor I are physicians. And so when we're having our discussion, we really are not in a position to provide personal medical advice. But, I think one of the more interesting things about your talk is that, I hope everybody understands this is a research project, right? You're trying to figure out, can we even do this?
Yep, that's right.
Right, and and what's remarkable is that it works. I mean, it's really convincing, and it's also clear that this is just the tip of the iceberg.
Absolutely.
So, I'm kind of getting the feeling that we're all basically about to be involved in this single large clinical trial where all of our information and patient records are going to be munged together to make some big prediction. But
that's
anyway,
definitely the way things are headed.
Yeah, exactly. Yep. So. I have some questions here, but I did want to offer the opportunity. Anybody in the audience here have a question, in-person audience? While we find somebody. So one of the questions that came up and a couple different people asked in different ways. You use 23andMe. And so, there's some concerns about, privacy for that. There's potential other sources. And it's also not really a complete genome either. And so could you maybe just talk about, the sources of information and where you see that going as you grow these sorts of studies?
So the original study is based upon the 23andMe data. It's not complete genetic information. So what they're actually doing is genotyping markers that have known genetic variation, and that's being used to calculate the scores. And it does provide most of the information actually for polygenic risk scores. But ultimately, I think the way things are headed is you'd probably get your genome sequenced at birth. That would be a part of your medical record. We have to figure out how to do that. Actually, it's not so simple in the United States, especially because we don't have sort of standardized systems. In Europe, they have more standardized sort of health record systems. But we're working on the technology or not me, but others are working on the technology to integrate disparate health information with genetic data. And ultimately, I think it would be sort of a on-demand sort of thing. You go to your physician, and just like they would check your cholesterol levels to determine whether or not you need statin therapy, that check would also involve checking your polygenic risk score that's already pre-calculated from whole genome sequence data that potentially is generated at birth.
So people are generally worried about artificial intelligence. And there's lots of public discussion. But for this very specific application, are there any dangers? Are there concerns? Like, what keeps you up at night? One of the things AI can do is dream. And so if you've got a patient that has a profile that isn't represented in your data set, it'll still predict something. So, yes. But what keeps you up at night about this?
So our model is not going to become conscious all of a sudden, so I'm not worried about that or any of the ethical concerns of artificial intelligence. Another reason why you mentioned some people might not be represented in the data set. I would say actually that the current models that we're using are based on averages, so we're not really represented well within the current models.
Yes.
The machine learning models, they are naturally able to generate risk in a heterogeneous population. They're not linear models. They have sort of branching, and different people have different risk profiles. So it'll do a better job actually capturing the heterogeneity of risk. But UK Biobank is essentially all white individuals. It's from England. England is generally white European individuals. So these models probably work best for white European individuals. That's probably the biggest gap, or what we like to improve, is the generalizability of these predictions to non-European individuals.
I want to ask you a little bit. So you're not a physician, but there were some physicians involved in this work. So we have all these great clinical scholars here at Scripps, and I know all these people on your team. And so what was their role in this work? Because it wasn't really in meeting with patients. It was really providing their expertise on curating the data that went into your models. But tell me a little bit more about how you involve clinician scholars in this data science project.
So part of the reason to involve them is to train them in the use of data science and genetics. So part of it is just their exposure to this information, so they can go back and use it in their own practice. Then they do help us with the curation as well. Let us know what's important, what's not, what we should include in our model, what we should not. But I think the biggest part of it is the last piece I showed, the interpretation of those different risk profiles. I didn't get into these details, but basically would get into whether or not their predictions match clinical guidelines, what's out there, and sort of what targets should be hit. And it turns out that the model is able to learn pretty well what the different targets are that different risk groups should hit. So they up front, they provide us like a sanity check in terms of what we should include in the model, and then at the back end, another really important sanity check in terms of interpreting those models.
Right. So another aspect of physicians is that, and our colleague Eric Topol wrote this book, The Patient Will See You Now. And people come in armed with all this information from WebMD. And so now you've got this other tool. And so part of the enterprise is making sure that clinicians are involved in adopting this as a welcome source of information.
Yes.
And so maybe just talk a little bit about that and how do you embed this new way of thinking into clinical practice?
Yeah, well, first I'd say they're going to have to because this information is out there, and although we don't tell people necessarily, I mean, we would like them to go to discuss it with their physician, but we don't tell them to go discuss it with their physician. They do. About 40% of the people within the MyGeneRank study at least intended to discuss those results with their physicians. And then the Peppers study, I mentioned that we're actually involving physicians directly in that study, so they are going to help recruit patients, and then there will be a set of surveys for the physicians that is directed around their understanding of the genetic risk information and what they did with that information. So we can learn from that to sort of learn how best to deliver the information in a way that physicians can understand it. And I think ultimately what you'll see is that more and more people will go to their physicians with this information. We develop a kind of a physician FAQ that they can look at as well. And to some extent, it'll be sort of a forced education to some degree with people just going to their physicians with this information and us doing the best that we can to arm those physicians with the information they need to make the right choices.
Great. Well, it's clear you're kind of on the leading edge of this, and it's clear that there's a huge wave of application of machine learning to managing our own health. So that's thank you very much. We're kind of over the hour, so I do want to just draw to a close, but I want to draw your attention to the next front row. So on November 29th, we have Xin Jin, and Xin is one of our young faculty stars, and she is developing gene editing approaches to address neurological conditions. And so thanks, Ali, and I look forward to seeing you all, and I will be right there in the front row.
Thank you. Thank you. Good job, man.